Hi, my name is Abby, and for my REU project at Dauphin Island Sea Lab, I am working in Dr. Titus's SimBio lab under Dr. Catheline Froehlich, looking at the adaptive functions of clownfish color patterns on different sea anemones in Papua New Guinea. There are 3 parts to my project. The first part I worked on this summer was stable isotope analysis. I processed fish and community samples that were collected earlier in the year in order to get their carbon-13 and nitrogen-15 stable isotope measurements. Basically, I dry the specimen, grind them up, weigh them, and fold them into tiny tinfoil cubes that will be sent elsewhere to be analyzed. Later this summer, I will use this data to measure trophic and isotopic niche overlap. The other 2 parts of my project involve analyzing fish videos to see how clownfish use different anemone species. I use ImageJ Fiji and the Aid Habitat Package in R to assess clownfish territory, looking at their estimated home ranges using kernel density. And this is my workbench. So the basic gist of this is that we have all these samples from Papua New Guinea from 2025, and these are community samples, and these are samples of anemones, and we're going to need the stable isotopes of both of those. So we're checking for nitrogen-15 and carbon-13. So what I do is I dry them out. So I dry the samples out in the dehydrator, and then I crush them into powders, and then I take teeny bit of those powders and put it in a tinfoil capsule, and then we send them off to Berkeley where they will do the stable isotopes, and then we get the results back and analyze it. Another cool thing is that whenever we have samples like this, so this is dried samples that we have already put in the dehydrator, and then I put ceramic beads that kind of look like this into these, and then I'm going to put it in this machine, which is called a bead ruptor, and then this is going to take the beads and just shake it very violently so it turns into a powder. And then it should be all done. Sometimes it doesn't get all of them, but these should all be powderized by now. So if you look here, that looks pretty powdery to me. Algae is usually the hardest thing to get